Hi everyone, and thank you for the kind introduction and the opportunity to share our data and what I think about using Miniscope in such a way that we can record the single cell neural activity in a freely moving mice, so we can have the behavior time lapse analysis. So as you can see, this is the classic view of the example of miniscope recording that an animal is behaving in a time-locked way, and we have single neural recording. And I'm gonna share what we have achieved and some tips that you may get from it. Okay. So the content's gonna be I'm gonna begin with the general question, the aim of the neuroscience and why miniscope. And the most of my time I will share with the behavior time locked analysis and maybe some technical tips later. For the aim of neuroscience, probably you all people are a neuroscientist trying to study neuroscience. So for me, neuroscience is to understand which neuron is responsible for certain phenomenon or certain behavior or certain cognition. So it would be easy if there is a single neuron responsible for eating. I'm an eating researcher, so that would be very easy if there's only a single neuron doing all cognitive part and all behavior part. But as you all already know, that is not the case. That most of the neurons are not doing one function, but they are in a pathway between the cognition to the behavior. And from this multifunctional aspect, there's tier one, tier two, tier three. So the first tier is the what, the identity. What this is? Is this an apple or it's a person or it's a cat or it's a chair? So to identify, we receive different kinds of the sensory modality: vision, taste, olfaction, touch, or uh, the sound. And there is multiple layers of the neurons for the cognition. So that's cognitive neuroscience. And there's tier two, that's real or affective value or something like a emotion. Is it something good or is it something bad? So is apple a good thing or is a cat a scary thing? So some of the neurons are doing their job by encoding their real or affective value. And there's the tertiary decision making and output. So what should we do? Toward it, approach to the apple, or run away from the cat? So we have to make a decision making, and and then we decide to do a behavior based on that, or autonomic neuroendocrine response. So uh, to understand a brain or understand neuron, we should be aware of this multifunctional, multidimensional aspect of the neuroscience. So for me, I started my lab like 10 years ago because there was a new tools for mostly for rodents, for mice. That I saw that the invention of this microendoscope, the main top uh, subject, and the fiber photometry, that we can do neural activity monitoring in vivo in a live, freely moving animal. I saw the invention of new technique, and there's another technique is optogenetics. And for me, this fiber photometry or microendoscope, miniscope, is like a mind reading. I can read your mind right now. You think you are thinking about a strawberry cake or something, and I can do mind writing or so I can make you do something. So the invention of these two tools, mind reading and mind writing, has really evolutionized the whole the neuroscience field. So to understand the technical limitations or technical function of these two tools will enable us to study the role of a certain neuron. So about what about the miniscope? So when you think about how you will do your neuroscience, you have to think about two things: temporal resolution and spatial resolution. So for the temporal resolution, we are talking about like how many millisecond temporal resolution do you want, or how many longer duration do you want. And so for that, the multiphoton microscopy or miniscope have a good temporal resolution within a second duration. That's for main purpose. And for the spatial resolution, you can go beyond like a nanometer or beyond centimeter. But for this tile, I think the single unit is about like 10 micrometers. So about this measure scale, time limits are important. So I think this miniscope is good for this spatial and temporal resolution, so we can have a single cell recording within a sub-second temporal resolution. So let me quickly go through the basics of the techniques. So we use the 𝗔𝗕𝗦𝗧𝗜 calcium indicator. So it shows green fluorescence in correlation with the calcium concentration. So that's a proxy for the neural activity. And we can genetically introduce this 𝗔𝗕𝗦 by using viral infection or transgenic, so the whole brain or whole body will have this indicator in their neurons. And for today's talk, I'm going to focus on the technique called miniscope that's using this lens-like lens so that we can image single neuron inside the brain by using this lens and attach a microscope on top of it. But for some of the talks, I'm going to talk about this optic cannula-based fiber photometry. So that measures the bulk activity, the sum or average of the 8 neurons at the same time. For this, I can distinguish neuron 1, neuron 2, neuron 3. It's a single neuron resolution study. So yeah. So this is the general structure of the miniscope or miniature microscope. Basically, it's a microscope that's small. So we can have this microscope on the top of the mouse head and let them freely move because it's not that heavy. So they can move around like a small hand. Yeah. But they all have this microscope functions of the lens and cameras. And the fiber photometry, it doesn't have this lens, so it only calculates how much light are within the tip of the optic cannula. So it's just it produces just a single number, the brightness of the whole 8 neurons. But for this, we can record the image of the 8 neurons or 100 neurons. Yes. Okay. So about the indicators, we can use calcium-based imaging or voltage-based imaging or electrophysiology. So for now, I'm going to focus on the calcium imaging because of the advantage of the cell type specificity of high signal to noise ratio. So the calcium imaging uses the cellular calcium concentration as the proximity for the electrical activity. But the voltage imaging, in theory, is more better because it directly records the electrical activity by the voltage membrane potential, but it has lower signal to noise ratio. So I believe somewhere soon people will invent a better voltage indicator with a higher signal to noise ratio. But for now, I think calcium imaging is better. For the electrophysiology, people have been using it for more than long ago. But the main disadvantage is that it cannot do the cell type specificity that easily, and so that's why I think the calcium imaging and using the miniscope has the advantage. So the calcium indicator uses calcium, and the signal looks like this, going up and down with a cellular uh, temporal resolution. And we can introduce this indicator by viral injection or the other ways, or transgenic mice. And we can image the neuron, this like 7 neurons at the simultaneously in the live, freely moving animal. So we can do this head fix, or we can do freely moving. So this is the general overview of the techniques. The fiber photometry, we only have one number, the total light intensity. So it's going up and down. So it's the sum of the like 100 neuron in the area. The single or multiphoto miniscope images this images. So there could be like 100 single neurons that we can distinguish. It's freely moving, and the 2-photon head fixed imaging is the same, but since we use a 2-photon, it has a better spatial resolution, so we can actually see the axons and like that. And so when you design your study, you should choose whether you will do a freely moving or head fixed. I prefer freely moving for my personal interest, because we can do more naturalistic, and we can have a longer temporal space, temporal distinction from one event to the next event. Yeah, so I think freely moving has an advantage in this way. But many people prefer head fixed, because you can deliver other cues or reward in a more controlled way. So it's the freely moving has the limitation of providing the cues or reward in a more designed way. But if you design in a certain way, then we can have the similar cue and behavior action sequence in a freely moving too. So I think one of the most advantage of freely moving compared to head fixed is we can actually see the mouse moving around, approach to the reward or moving away from the reward or threat. So this locomotion of a mouse gives us more data, more insight by using this freely moving design. So yeah, in theory, I think freely moving have a better advantage. Yeah, for those two reasons. We can have the locomotion part, and it's more naturalistic. Yeah. Now you can measure the motivation of the mouse using freely moving much better than head fixed. So when you combine these two techniques together, there is the new technique that's emerging is freely moving 2-photon miniscope. So the 1-photon freely moving is widely used. I'm going to show most of data using freely moving 1-photon, but the imaging quality is not so good. Head fixed, the 2-photon microscope has better resolution because of using this focal plane using multiphoton. So it has better imaging. But the Bruker or other companies are making new devices so we can do this high spatial resolution 2-photon imaging in a freely moving pattern, freely moving design. So that's please wait for these new techniques to be more widely available. So when you compare these techniques, the fiber photometry with the single photon or 2-photon, or mostly it's head fixed. The most important thing is the cellular resolution and the subcellular resolution. So I'm mostly interested in the cellular, not subcellular. That's just my personal preference. I'm not that into the axon or dendrite imaging yet. So for the cellular resolution, I think single photon is good enough for right now. But I like to use the advantage of the freely moving, and the cost and the skill required increase as you go through this way. And when you expand more, as I told you, electrophysiology has been more used, but there is the limitation of the molecular specificity or cell type specificity is limited. I think that's the major reason that I do not prefer the electrophysiology, and I prefer this miniscope because we have the cellular specificity and molecular specificity within 10 millisecond temporal resolution with some setup costs that I can manage to use. So it depends on whether you use the DIY open source or the commercial devices. 2-photon is much more expensive. Yeah, and this is the general design of the optical path for the photometry. So we can do multi-photometry too. And you can use this technique with two color, or you can combine with the crimson optogenetics and do stimulation and the photometry at the same time. And this is the miniscope. Yeah, as I have told you, you can miniscope like 100 or more neurons at the same time using this lens-like structure. And so recent techniques in the 2-photon imaging with the single cell stimulation, you can selectively stimulate the neuron at a time. So this is another technique. Yeah, I'm not gonna go through that. So for the cellular imaging, the initial image may seem a little blurry, but the Bruker or other companies have, or the open sources have this software. You can do motion correction or cell detection and single cell extraction, and then you have much better image and much better signal to noise ratio. And then you choose your region of interest, and you can have single cellular imaging on your data, and you align them with the event of the cue or event of the behavior. Yeah, and then based on this event based analysis, you can cluster your neurons. So yeah, this is another nice picture of the inscopic miniscope. So you can have this miniscope and some version of the miniscope invoke provides optogenetic stimulation too. So you can optogenically stimulate this axon while imaging these green cell bodies. And you can align them with the leaking event. So the basic data simply looks like that. So you have image of this like 20 or 50 neurons at the same time, and you can have this neuron number 1, neuron number 2, and then you align the neurons and have your data, and you align them with the your event and analysis. And they also have this ready to image virus. So yeah, you use this virus and you can infect mice or rat neurons. And there is another integrated lens, that the lens and the base plate is integrated at the same time, so it can lower the surgical burden. Yeah. And for the calcium imaging analysis, I told you you get the image acquisition with the time lapse, and you do motion correction and neuron the identification, and you drive the action potential detection and registration. And the time required for the experiment is somewhere summarized like this. So you develop your animal protocols or materials, and you inject the virus and implant your lens and fibers, and you optimize it, and then you record your signal and you check the signal. If it's not appropriate, you go back and you optimize your virus dilution or the targeted techniques, and go back. Yeah. So it requires several months to really set up the experiment. And once you set up, then you are good to go to acquire your imaging data. So that's, yeah, the beginning. And let me do the main part of the talk. So I'm gonna tell you about how we use them in a certain way, so we can have the behavior time-locked analysis. So as I told you, when you design your experiment, you should think about these components. So what will be the readout of the behavior? Is it a level or sensor or leaking or any position or posture? Will you do something innate or something acquired that requires learning? So, and you choose head fixed or freely moving. Will there be a cue or no cue? And you set up your behavior paradigm based on your design. And as I told you, you think about uh, uh, what would be your hypothesis that if you choose certain neuron, like in visual cortex, you have to think about, oh, it's gonna be something to do with the stimulus identity. And if you do something in like a lateral hypothalamus, that's my favorite place. Then it could be something like a behavior outcome. Yeah. So if you do something in the amygdala, then you should have your prior hypothesis that is something that has to do with the reward or affective, emotional value. Yeah. So you make your hypothesis and then design your behavior. So for me, I have this thought in my mind that there is the environment, sensory, brain, unconscious, and conscious level ideas, and then your brain starts a motor behavior, and then it changes the environment. So for me, I'm doing eating or homeostasis research. My best interest is that when there is a deficit in the nutrient, your brain, the homeostasis center calculates the need, and then it drives the motivation, craving, wanting, and you make a decision based on this conflict. And when you decide to do a behavior, so there is a time for the craving ramping up. I want to go, I want to go, I want to go, and then you start to go later, and you find something, and you make another calculation in the brain, and you start to approach. And when you get close to your viewers, like you enter a McDonald's restaurant or a pizza restaurant, you suddenly feel the smell of the bread, and then you decide to consume, put it in your mouth. When you start to put it in your mouth, you taste the sweetness or saltiness, and then you experience the pleasure. Oh, it is so delicious. Yeah, the pleasure is at this time moment, and you keep eating it, and like 10 minutes later, you feel, oh, I'm so satiated. I feel so full. I will stop eating. So this is the behavior layer that we can measure in the mouse. But inside the mouse brain, these multi functions are happening in the brain, and I think there is every neuron responsible for this. And then after a single event, your brain memorizes the event. And the next time when you go there, like 1 week later, based on your memory, you make a different decision because you have different calculation based on your memory. Yeah. So, and this cue also drives your motivation. So by understanding the structure of the cognitive, emotional, behavior components, I think you will have a better design of your experiment. So yeah, you do something before and you start to forge for your something, and if it's not good, you forge again, hunt again. But if it's good, then you start to ingest and feel and when you stop eating, you go to your next behavior, yeah, meeting your girlfriend or something. And yeah, so in the brain, there is the cognitive center, that's visual olfactory or gustatory center, and there is the memory center. So memory center like hippocampus tells you that's a strawberry donuts, and your hypothalamus or brainstem let you know that you don't have enough energy, and your reward-based decision-making centers like VTA or nuclear accumbens, they make a decision whether go, no go, approach or avoid, approach, avoid, approach. Should I go? Should I go? Should I not go? Should I go? Should I not go? So there could be like a 10 seconds of your mind going up and down, but no behavior. And then when you choose to behave, you start to move toward the McDonald's restaurant. So there's a temporal distance between your reward-based decision making and your behavior. But yeah, it's so difficult to measure this. But if you design it well, you can measure that. So in our lab, we try to investigate the desire mostly. So where is the desire in the brain? So yeah, if you have a desire, you may have desire for strawberry donuts right now, but you don't have any behavior yet. But so later, like 1 hour later, you can go to the donut store and at that time, point is when the behavior is evident. Yeah, we can measure the behavior. So we've been studying desire and hunger, the upstream of the desire, and the palatability using mouse and human and monkeys. And let me show you the mouse data. So I told you about the design. So you have to think about the design. So in my view, all animal measures their state, and if there's a deficit, your brain calculates that you need something. You need like 500 kilocalorie of any food. Yeah, you can eat pizza or you can eat cake, but I'm gonna eat hamburger. So I have a motivation for hamburger, and then that drives the behavior toward the hamburger. And when you start to put it in your mouth, yeah, you can approach to the hamburger, but when you put the hamburger in your mouth, you feel the pleasure, and there's like a 10 minutes of the cycle of the, oh, so good, I want to eat more, so good, I want to eat more. And then 1 hour later, your body digests the hamburger and your glucose is coming back up. So I think these two are the cause of the behavior, and these two are consequence of behavior. And always brain have this sensory cue driven prediction based on this. So I suggest you to have this some kind of framework of all the components of your interest. In our lab, we have been trying to investigate each components by studying them, and Miniscope have been one of them our main tools to investigate this neuronal component responsible for this psychological components. So yeah, when you record something, yeah, eating and neural activity looks like this, and you say, oh, this neuron is activated, increased by eating. But there are multiple ways that can happen. So these neurons are activated after you start to eat. These neurons are activated before eating, but inhibited at a few milliseconds before eating. And you can have sustained activation or something like this or something like this. So a researcher can say that all six are the same thing. It's increased at eating. But if you have more data on the cognition and behavior, so if you can have more data on the event about the cognition, when they see or smell the food, and when they start to move, or when they start to put that in your mouth. So if you have more data, you can have a better insight about your neurons that you recorded. Yes. So it has been suggested in this nice paper in Neuron that the sequence of a trial event is really important. Initiation, action 1, state Q1, action 2, outcome Q, outcome. So this action, outcome Q structure sequence is really important. And you can do more dopamine recording, or it's a real trial, omission trial. And you can do this analysis to and if this action 1, action 2, or outcome are aligned with the neuron that you're recording. So there is another possibilities that this neuron can be encoding outcome or action on value or the value state or yield rate or something like that. So if you think deep and try to design in a way that you are using this sequence of trial events, you have better analysis. And there is another important aspect is that if you have this could be the brake or the accelerator, because you cannot tell whether this neuronal activity is increased is the cause of the behavior or like a feedback mechanism for the behavior. So if you were to record the accelerator gas or the brake of a car, then when the car is parked, there is no activity. When you drive in the highway, you press both lever frequently during your driving to the highway, but when you stop driving and park again, they are all decreased. So this higher activity could be the gas or it could be the brake. So the way to test this is to use the causality testing is the optogenetics. So optogenetics will let you distinguish whether it's the cause of the behavior or not. So, and I'm gonna suggest another way of thinking is normative approach is something that you think about what control the animal should have for optimal survival. So what is the ideal brain working? What is the principle it should follow? So it's like building a computer, then you will program it a certain way so that the machine or the animal performs the best optimal or normative way for best survival. So I've thought about this, and need and motivation, so hunger or appetite to eat, have different functions. I think it needs, I need 500 kilocalories like ordering for change for the future. But for the motivation is the telling you to action right now. So can be a situation when you don't have need, but you have the motivation, like when you want to eat dessert, when you don't need any calories. And there is a circumstance in like a war, you need calories, but you don't feel any appetite. And so this is the feedback inhibition, that you do something and your behavior inhibit your thing. But there's also feed-forward regulation that's less well known, and it's like a pressing a brake before your stop sign. So when you see the stop sign, you start to press the brake before your actual outcome, so that to have you stop your car before the stop sign. So it has been well known because of the thirst and the hunger. When you record a thirst neuron or hunger neuron, people thought that putting in the gut will decrease the thirst neuron or hunger neuron faster. But on the contrary, oral ingestion, when you put the water or food in your mouth, these neurons are decreased, inactivated much earlier than putting in the gut. So it's feed-forward satiation signal by the taste or smelling on the side of the cue. It makes more sense because if you start to drink the water, your thirst decreases, so that you will not drink too much. If your thirst persists until you have the dilution of your blood, you will drink like 2 liters, too much. So to prevent the over consumption of water, your thirst are feed-forwardly satiated by this sensory. Yeah, so I have thought about this, and for the energy, when you have enough energy here, then you don't have any need or you don't have any motivation. But as soon as you go on and you deplete your stored energy, you don't have enough energy, so you have high need. But since you cannot do anything, there's really no motivation to do anything. But once you have access to your food, then your motivation starts to increase a little, and then your need decreases because you will predict that you will get something soon. And as soon as your motivation reaches a certain threshold, your behavior initiates, and then you move to your goal. And as you finish your goal, everything ends. So this is the model that I suggested, and we made a computation model for this. And it has been suggested that this, in addition to this feedback regulation, there is a feed-forward regulation that prevents the error, too much overconsumption. So sensory cue inhibit the hunger neuron in the dorsal medial hypothalamus. So that was the basic idea. Because of the important research that if you measure this HRP neuron, people thought that this would persist until you finish your eating. But 10 years ago, this paper showed that as soon as the mouse see or smell the food, this HRP neuron are inactivated even before eating behavior starts. So HRP neuron cannot be the direct driver or motivation for the eating behavior. So based on this thought, people thought that, yeah, these HRP neurons are inhibited by the food discovery. And then there's some other signals that really drive the eating behavior. So this is our the conceptual framework that we are based on. So let me go over this talk about the lateral hypothalamus neuron. I think they are encoding the motivation. So we show that there's two subpopulation of the neurons. So I'm going to go through that. Yeah, I've been studying this, and I suggest that craving, seeking, and consumption are three different phases of eating. And 10 years ago, Stuart's lab showed that in the lateral hypothalamus, there is the consumption neuron and the appetitive neuron separately. Let me show you the data. So they use this freely moving miniscope and nose poke and lick, and they are measuring this several 20 neurons at the same time, and measuring which neurons are encoding the nose poke or which neurons are encoding the lick, comparing this behavior paradigm. It's like, they said that there's consumatory cells and there is the nose poke appetitive cells, that you align them in a behavior, you can distinguish them, and among the neurons, there is the nose poke excited cells or consumption excited cells. Yeah, that was their basic thought. And we thought that, this lateral hypothalamus 𝗪𝗏 neuron, among them is the leptin receptor neuron. And people have suggested that leptin receptor neuron lateral hypothalamus is the driving the feeding behavior. So we look for the neurons and the lateral hypothalamus, the leptin receptors are there. And we found that there is a leptin receptor neuron in this area. So we chose to study them. And this was very important to us, that we tried to develop novel new phase-specific experimental paradigms. In a certain way, we can distinguish this all different phase of the event. Like I told you, the sequential event or cue or behavior sequence design is important, so we can have enough temporal distance between this event so we can distinguish the neuron at the same time. So this is one example. This mouse is searching for the food. The food used to be here, but not anymore this time. So reach for, there should be food, where's the food? Oh, forget it, there's no food. So when the mouse stop reaching for the food, this neuron goes down at the time. So time-locked behavior recording. And we have done this neuronal recording of the 𝗪𝗏𝗔𝗟 neuron, the total population, the higher the population, and we saw many 𝗪𝗏𝗔𝗟 neurons, and some of the neurons are increased by the consumption, but some of the neurons did not move. So we're trying to map total neuron and the food-specific neuron. And I will just show that when we investigate the total lateral hypothalamus 𝗪𝗏𝗔𝗟 neuron, only minor % of them are food responsive. So this is the food trier, non-food trier. So most of them were non-specific. But when we limit them in the lateral hypothalamus leptin receptor neuron, most of them were food specific, and only minor of them were non-specific. So we can show the subpopulation by using this miniscope. And this is photometry data that suggests us that we can have this analysis. So this mouse is going to eat their food reward at the end of the this maze. So this is the first time the mouse is moving toward the food. So we are recording the lateral hypothalamus leptin receptor neuron, the bulk neuron, this time photometry. So the neural activity did not go off until the mouse discovers the food. So this is a non-goal directed locomotion until now. And as soon as there is a food at the end of the corridor, and when they find the food and approach the food and start to eat the food, it goes up. So we could see that neuronal activity goes up before the contact. And more interestingly, when we train the mouse so that the mouse knows that food is at the end of the corridor, so we have a more interesting event when you open the door. This mouse now knows food is there, so run for the food and goes there. So at the same corridor, now the neuron activity goes up at the beginning of the seeking. So we could show that after learning, the door open, the accessibility will increase the neural activity. So we could know more things about this neural thing. So, and then we did another interesting study. There's no door, so the mouse have to decide when they want to go. So we could see the ramping of the motivation before the behavior. I will show you again. So should I go? Should I go? I wanna go. 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 And let's go. So there could be like 6 seconds delay before when they have the motivation go up and the behavior starts. This is another way of showing that. And for using the miniscope single cell analysis, we derived this paradigm so that we compare the food trial versus no food trial, so we can distinguish the neurons which are responsible for the consummatory behavior. And the data show that, this mouse is trying to move. As soon as it goes to food, these green neurons are activated. And as soon as the mouse starts to eat, these green neurons goes down and the purple neurons goes up. So that's distinguishing two population by temporal dynamics difference. More interesting, we'll do no food trial, but the mouse doesn't know that food is not there. So expect the food and run for the food. So the same green neuron goes up, but no food, no food, no food. So when there's no food, the same purple neuron are not activated. We can separate these two neuron population in this single cell recording. So when you compare seeking, consumption, seeking, consumption, seeking, no consumption, seeking, no consumption, these consumption neurons are activated only if there's a food, but the seeking neurons are activated anytime you do seeking. So we can distinguish the seeking neuron versus the consumption neuron. And the single cell analysis also show that, the seeking neurons are activated at all trial, no food trial, but consummatory neurons do not activate by the no consumption trial. So by using this behavioral time lapse technique, we can distinguish these neurons. And we do this trial like 5 times and show that this is very reliable. Every time it goes like this. At a similar time, there was another report studying the same lateral hypothalamus leptin neuron, and they showed this data of their time lapse activity. But I want to tell you that I think they should analyze the data in more detail, because they say this is for the inhibited neuron, but in my view, these neurons are the orange bar is when they start to eat. So it may seem to be inhibited by the eating, but there's a slight increase and decrease like a few seconds before eating, just like our data. So you can say that this is for the inhibited, but you can also say that that's activated by the food seeking, as we have been shown. So this is just example to show that if you know more behavioral cognitive event data, you can distinguish the function of the neuron in better way. And we did optogenetics to actually stimulate the neurons and see if they have the causality effect. This is causing the food seeking, and this is causing the food consumption. We could increase the food consumption. So this shows you the causality, and we can do inhibitor to inhibiting optogenetics to show that it has the function. And we have another example. So it's about the cognition part. Let me go quicker. So pre-ingestive sensory satiation. When you see something, it can have satiation. So in this trial, we use the same similar paradigm. And when we do bulk photometry, we could also see that these neurons are activated after conditioning at the food-seeking moment, similar to the previous report. So this time we are measuring different neurons, dorsal medial hypothalamus GFP receptor neurons, and we do miniscope again. And in this miniscope, you could dissect this mouse. Okay, should I go? Let's go right now. So we can really time lock the seeking star, and we could time lock the consumption star and record a single neuron at the same time. And some of the neurons are activated before consumption, some are activated at the consumption start. So that's two different neurons. And later, we have another no consumption trial too. So this is the same mouse. So when we open the door, the mouse starts to move towards the food. So the same frequency neuron starts to go up. But when they get there, there's the food. But somehow this time, the mouse decide not to eat the food. So no consumption. So there is no consumption neural activity. So by comparing this, we can dissect the neurons. And let me skip this. Yeah. And we did optogenetics to show that they have the power to increase or decrease the satiation. So by doing this, we could show that, the previous HRP neuron, lateral neuron, and the upstream of this neuron by showing this. And I have one more example. Let me go quicker. So it's about the memory acquisition neuron in the nuclear accumbens. And we want to record the memory acquisition neurons in the nuclear accumbens. So we record this neuron, and we could see that they are activated by this. We are recording the nuclear accumbens MPY neurons. It's unpublished data, and they are activated by the hyped diet. And we could show. Let me show you this video first. So before conditioning, this red zone doesn't mean anything. So the no neural activity is recorded by this. But after they eat the sugar 3 times at this red zone, and then when they after the conditioning, they expect the sugar in the red zone. So by this expectation, neuron use goes up. So we saw this thing, and we tried to do the miniscope later, and so we are recording the single neuron at the same time, and as they expect, they go and eat the food, and they say, oh, there's the food. So we could distinguish 2 different neurons at this time. And after running more, and at the omission trial, this same purple neuron goes up, but since there's no food, so this current value neuron are not activated. More interestingly, we analyzed this omission trial 5 times. And so this is the omission 1 and omission 2. Oh, I'm gonna go there again. One failure is not good for me. I go there, oh, no food again. So this is failure 2. And they'll go to their failure 3. So even though the mouse failed 2 times, they go there again. And we can compare these 2 neurons. This neuron activity goes up regardless of failure, but this neuron, every time it fails, the neuron activity goes down or lower. So I think this neuron do not update the new information like habit-like, but these neurons are updating the new information like a goal-directed or learning. So we could distinguish the inflexible or flexible neuron, and we can track the neuron for pretest next day or next day and see if the same neuron in this upper right, left upper corner, and they are activated by a certain time point. So this is the longitudinal follow-up of the neurons. And we also did optogenetics to show that this neuron have the causality effect on the behavior. So let me quickly end by the technical tips. So mostly we use the 6S, the slower type, but you can use the faster type for different temporal dynamics. And there's another set of the 𝗔𝗕𝗦. You can choose 8S. That's more sensitive than 6S, but the temporal regional, you can choose the temporal regional too. And this is a summary of the different methods. And for the multi-photon miniature microscopy will be, I think, will be very popular soon. And for this, I want to just show you that we use this multi-photon calcium imaging, and there are different softwares, but there are more software or pipelines that are coming up. So you should be aware of this or analysis. And about the lens, I show you most of the lens look like this. But if you use this pressure lens, you can image area in this vertical way, not only the horizontal way. This is another thing to consider. So thank you for the attention. So I have some slides. So if you have any questions, I would like to, happy to answer them. Yeah, thank you.
Yeah, thank you, Dr. Choi, for your great talk. I have a question regarding the 𝗡1 neuron, because 𝗡1 has been very popular in the diabetic and also using weight research. Do you think the mechanism that you found in rodents are also conserved within other mammals, especially human beings?
You mean the GLP-1?
Yeah.
Yeah. For the GLP-1, that's an important question. Yeah, thanks for bringing it up, because I had the slides, but I just wanted to have some Q&A time. So for this talk, we are measuring the dorsal medial hypothalamus and GLP-1 receptor neurons, and showing you this data. As you asked, we wanted to know if they are same in the humans too. So we did human study. This is the human behavior study. So this is the actual scene of the human behavior experimental study. We are giving them chicken, and we have a temporal sequence of the baseline, and this pre-oral pre-ingestion phase is when you see or smell the chicken, but do not put it in your mouth. And we tell them to put the chicken in your mouth. So this is oral pre-ingestion phase, but do not swallow. So chew the chicken, enjoy the chicken flavor, chicken taste, the sweetness or saltiness, but don't swallow. And after a few minutes later, let them swallow. So there's 4 different phases, and we recorded their satiation score by asking a survey, a number, visual analog scale survey. How much satiation do you feel? Score of 9 or 10. And then we saw that when they had this drug injected, their satiation score increased at this pre-ingestion oral phase. That's when they experience food in the sensory way, but there's no swallowing. So we could see this increase in the neural activity, and we did the fiber photometry of the same neurons, and we could show that the neural activity is much faster increased when they have the drug injection. So before injection and after the GLP injection, the neural activity of this population are much sensitive to this event. So I think these 2 data mirrors. This one is the neural response to an event, and this is the symptom response to an event, and they show similar temporal dynamics. And we propose that this is the evidence that in humans that this symptom of the increase in the satiation is encoded by this dorsal medial hypothalamus GLP-1 receptor neurons. Yeah, so and we also did brain human brain tissue antibody staining IHC to show that there is this receptor in this area. So I believe this phenomenon is also valid in human. I think it would be better if we did the neural encoding of the humans too, using the same behavior paradigm. But at this time, we did not do that. But we have ongoing study to show that, recording the neural activity.
Yeah, thank you. Yeah, I have a second question. That's regarding the mechanism that you propose, because it's based on your model. GLP-1 neuron, it seems on the most upstream of the whole neural circuit. Yeah. So do you think that's actually the initiation center for the whole feeding behavior, or is any other inputs also may regulate its upstream or regulate the ARC?
Very important question. So I believe that there are two parallel pathways. Yeah, one pathway is the homeostatic pathway. So that's based on their energy deficit. So HRP calculates how much food, energy, or calories you need based on your deficit. So this HRP neurons are driving this appetite or craving by lateral hypothalamus area neuron, and that's driving this behavior. That's the homeostatic pathway. And this dorsal medial hypothalamus GLP-1 receptor neurons are one of the upstream of this homeostatic pathway that inhibits this HRP neuron even before starting to eat, based on this cue prediction to prevent the over consuming or error. That's the all homeostatic pathway. But I believe there is another pathway that's not using this HRP. So when you had your lunch, you know you are full, you don't need any more energy. So HRP is very low at this time point. But when you see ice cream or when you see chocolate cake, and there is another higher cognitive reward-seeking hedonic pathway is hijacking this lateral hypothalamus neurons, so that you will start to eat your ice cream even though you don't need anything. So yeah, there could be two parallel pathways. Yeah, one for homeostatic, eat for survival. There's another pathway, hedonic pathway, eat for pleasure. So there's two pathway convergence, and mostly we studied the homeostatic pathway for now, but my later talk was trying to investigate the hedonic pathway. Yeah, thank you for the question. And the Miniscope, it has been the best tool that we can distinguish this neuronal component by using this time-locked, event time-locked or behavior time-locked way.
Thank you. My last question would be, for studying the homeostasis, based on the current technology limitation, what kind of a tool would be the dream tool that you think can really conquer the major question with this field?
Yeah, I think dream tool. Yeah, for me, I showed you a little video earlier. Yeah, that for 7-8 years we have been trying to use the one photon miniature microscope, but I think there soon, yeah, right? Hisham, soon you will have this freely moving 2-photon microscope, right? So
yes, it's going to be released
soon, right? This year, right?
this year, very soon.
Yeah. So I have enjoyed this freely moving one photon for like more than 5-6 years, publishing good papers. But we always had this problem with the resolution of the image, and since it's not the focal plane is not exact, so that's one photon imaging that is just accumulation of the whole image, so we cannot really dissect in the G stack, right? So but using this multi-photon 2-photon really moving miniscope, we'll have a better imaging resolution with the G stack, so we can have a better single cell identity about their positions, and so in the G stack too. So if we have this freely moving 2-photon miniscope, I think we can do improve the science in two advantage way. One is because we have the better imaging quality, we can improve the longitudinal follow-up success rate. Yeah, because of using this blurry image, it's really difficult to see which neuron is which neuron, especially if they are in the same G stack. It look the same. So, but using this 2D photon, we can do better in the longitudinal follow or have more neurons. And a second advantage, I think, is that since we have the 3D structure of the single neurons, like 50 neurons scattered in the 3D space, and we know the exact location of the 3D population. And we can, after doing calcium imaging for several days or weeks, when we sacrifice the animal, we get this cubic brain sample, and then we do 3D imaging of this same 3D cubic sample, and we do molecular functional identity imaging using FISH or other techniques, and then we can really tell, oh, this neuron has a 𝗔𝗕𝗏 receptor, this neuron has a leptin receptor, this neuron is dopamine neuron, and this neuron is 𝗙𝗏𝗟𝗔𝗟 neuron or double positive neuron. So someone calls it karma, yeah, the calcium imaging and the molecular imaging later. I think that when we combine the two-photon miniscope with the functional, the molecular imaging, then we'll have the functional identity and molecular identity linked, and then that will answer much more questions. We can do this with the head-fixed two-photon right now, but most of my study requires this fully naturalistic freely moving. So we can have naturalistic emotion change during their approach and early detection and omission. So I really need this freely moving two-photon miniscope to advance the science. Thank you.
Thank you.